So meiosis events, that is meiosis I and meiosis II. So in meiosis I, prophase I, metaphase I, anaphase I and telophase I. Similarly, in meiosis II, prophase II, metaphase II, anaphase II and telophase II. So these are the events occurs in meiosis. So first one, in the meiosis I, prophase I. Prophase I of meiosis I is typically longer and more complex when compared to prophase of mitosis, because various substages are there that we will discuss later. Here, based on the chromosomal behavior, this phase is divided into 5 substages, namely leptotin, second one zygotin, third one pakitin, fourth one is diplotin, and the last one is diakinesis. So first one is leptotin. So in leptotin stage, the chromosomes becomes gradually visible under microscope. Now see the diagram. Initially, the chromatin material is like this. This chromatin material is gradually transformed into a rod-shaped chromosomes. The phenomenon is called condensation. This chromatin material becomes chromosomes that clearly appear in light microscope. See the chromosomes here. So this part is cell wall and cell membrane, and this is nuclear membrane. So this is nucleolus, and these are chromosomes, and this is normally cytoplasm. So in leptotin, the chromosomes become gradually visible under light microscope. So the compaction of chromosomes continues throughout leptotin stage. Here compaction means chromosomes comes just nearer with each other but not fused. That stage is called bucket stage. So these are the changes occurs in leptotin. Next, second one, zygotin. So it is the second stage or second substage of prophase I. Now here, see the diagram. Chromosomes starts their pairing. Chromosomes starts their pairing. So this pairing is called synapses. So see the diagram. Two chromosomes are just fused with one another. So these pairing chromosomes are called homologous chromosomes. Two chromosomes exactly similar in the size, shape and structure is called homologous chromosomes. So this pairing of homologous chromosomes, that is synapses, is due to a complex structure known as synaptinemal complex. So this synaptinemal complex is a special type of protein. This synaptinemal complex work like a zip-like fashion. So as a result of synapses, a bivalent is formed. That means, suppose in onion plant, so ileum cepa diploid cell contains 16 chromosomes. Here 16 chromosomes are fused to form 8 bivalents. That means 8 pairs. See the diagram. These two chromosomes are fused with another. After fusion, the chromosome is like this. So each chromosome contains 2 chromatids. So these are 2 chromatids. So for better understanding how 2 chromosomes are like this. So this is bivalent. One chromosome contains 2 chromatids. So 2 chromosomes contain 4 chromatids. That's why a bivalent contains 4 chromatids. That is tetrad of chromatids. Tetrad means 4. So that is changes occurs in zygotin. Next stage, pakitin. So in pakitin stage, the 4 chromatids clearly appear, tetrad. Now see the diagram. Is a 1 chromosome 2 chromatids. Other chromosome 2 chromatids. Total 4 chromatids are there. Here bivalent chromosomes are clearly appear. Bivalent chromosomes clearly appeared. It is long-lived stage than leptotin and zygotin. And the important change in the pakitin is appearance of recombinant nodules or chiasmata. Now here see the diagram, this point. The two non-sister chromatids contact physically, that is called recombinant nodules or chiasmata. For your better understanding, so it is one chromosome and it is other chromosome. So 2 chromosomes are there. So these points, recombination nodule points are chiasmata. So generally in the chromosome, DNA is there. So once chiasmata occurs, see that the chromatids of the same chromosome is called sister chromatids. That is 2 green colors are sister chromatids. Similarly 2 red color are sister chromatids. But the chromatids of opposite chromosomes known as non-sister chromatids. So these chiasmata present between 2 non-sister chromatids. So as a result, crossing over occurs between two non-sister chromatids. Crossing over means exchange of genetical material between homologous chromosomes. So this crossing over also enzyme mediated process. So that enzyme is recombinase. So it is enzyme name. So as a result of exchange of genetical material, origin of new species leads to evolution. So in meiosis, gametes are formed. Before the formation of gametes, exchange of genetic material between two homologous chromosomes takes place. Again, male gamete fused with the female gametes, zygote is formed. Zygote develops a new organism. The new organism definitely shows a new character when compared to parent. That means origin of new species that leads to evolution. So because of crossing over, the packaging stage is most important stage of meiosis cell division. So that it changes occurs in packaging stage. Next, diplotin. So this stage recognized by dissolution of synaptenimal complex. Now see the diagram. So these two fused chromosomes try to separate because the synaptenimal forces becomes weak. That is a dissolution of synaptenimal complex. So as a result, bivalents separate from each other except at the sites of crossing over. That means except at the chiasmata point. Now see the diagram. Here between two chromosomes gap is there, but at some places fusion is there. So once dissolution of synaptenimal complex, here the X-shaped chromosomes clearly appear. So X-shaped bivalent chromosomes clearly appear. So that is a changes occurs in diplotin. But in oocytes of amphibians or oocytes of some vertebrates, diplotin can last few months or few years. So it is exception points in oocytes of some vertebrates. So that is change occurs in diplotin. Next, diakinesis. Now see the diagram. In diakinesis, the chiasmata move towards a terminal point. Terminalization of chiasmata. And second changes. Now see the diagram. The nuclear membrane gradually degenerates. Nucleolus also disappears because it is a end of prophase 1. Nuclear membrane and nucleolus disappears. Chromosomes are fully condensed. Here termination clear picture is there. Meiotic spindle is assembled to prepare the separation of homologous chromosomes. Just preparation starts, but completed in metaphase starts in diakinesis. So this stage represents transition to metaphase 1. So that is changes occurs in diakinesis. So to this prophase 1 is completed. Next, metaphase 1. So in this phase, bivalent chromosomes align on the equatorial plate. Now see the diagram. So with the help of spindle fibers, these bivalents are on the center of the cell. So that is equatorial plate. The microtubules from the opposite poles of spindle, that is spindle fibers, attached to the pair of homologous chromosomes. Homologous chromosomes, that means bivalents. Bivalent chromosomal congregation. So still two chromosomes are fused with one another at the terminal point. That means chiasmata are terminal points also. So that is changes occurs in metaphase 1. Next, anaphase 1. The homologous chromosomes separate completely. Now see the diagram. The bivalents are separated. The separated chromosomes move towards respective poles. That means bivalents are separated. Sister chromatids remains associated at their centromere. So this is the stage responsible for the meiosis is a reductional division. That means diploid cell is converted to haploid cell. The precursor stage is anaphase 1. That means bivalent dissociate into two haploids. One haploid move towards one pole, other haploid move towards other pole. In this stage also, one chromosome contains two chromatids. Here only bivalents are separated, but chromosomes do not become doubled. That is important point. Next, telophase 1. Here the changes. The nuclear membrane and nucleolus reappear. This is a nuclear membrane and the small point is nucleolus. This is a nucleolus, this is a nuclear membrane. So as a result, two haploid nuclei are formed. Now here, chromosomes undergo some dispersion. That means decondensation. only less decondensation there, because it is reverse to prophase 1. In the prophase 1, chromatin material undergo condensation to form chromosome, whereas here chromosomes undergo only some dispersion or decondensation in telophase 1. Next, after telophase 1, again meiosis II is there. The stage between two meiotic divisions is called interkinesis. Generally, it is short-lived, means the time is very less.